Then next include some other aspects related to monoclonal antibody. They are antibodies that are made by identical immune cells that are all clones of a unique parent cell. Monoclonal antibodies can have monovalent affinity in that they bind to the same epitope. The method relies on fusing B cells from an immunized animal, typically a mouse, with an immortal myeloma cell line and growing the cells under conditions in which the unfused normal and tumor cells cannot survive. The resultant fused cells that grow out are called hybridomas. Each hybridoma makes only one immunoglobulin derived from one B cell from the immunized animal. The antibodies secreted by many hybridoma clones are screened for binding to the antigen of interest, and the single clone with the desired specificity is selected and expanded. So the products of these individual clones are monoclonal antibodies. Then types: naked monoclonal antibodies. There is no drug or radioactive material attached to them. Then conjugate monoclonal antibodies, that means conjugated monoclonal antibodies. Monoclonal antibodies joined to a chemotherapy drug or to a radioactive particle are called the conjugated monoclonal antibodies. Then bispecific monoclonal antibodies. These drugs are made up of parts of two different monoclonal antibodies and they can attach to two different proteins at the same time. Then generation of antibody diversity. In the absence of antigen stimulation, a human can probably make more than 10 or to the power of 12 different antibody molecules. It's pre-immune antibody repertoire. Moreover, the antigen binding site of many antibodies can cross-react with a variety of related but different antigenic determinants, making the antibody defense force even more formidable. The pre-immune repertoire is apparently large enough to ensure that there will be an antigen binding site to fit almost any potential antigenic determinant, albeit with low affinity. After repeating stimulation by antigen, B cells can make antibodies that bind their antigen with much more higher affinity, a process called affinity maturation. This antigen stimulation greatly increases the antibody aerosol. Antibodies are proteins and proteins are encoded by genes. Antibody diversity therefore poses a special genetic problem. And antibody genes are assembled from separate gene segments called the B cell development. B gene segments during B cell development, the specific variable region and constancy region coding sequences that the tumor cells use to were present on the same DNA restriction fragment in the tumor cells, but on two different restriction fragments in the embryos. This showed that DNA sequences encoding an antibody molecule are rearranged at some stage in B cell development. A smaller number of exons encoding the C region during the development of a B cell, a complete coding sequence for each of the two antibody chains to be synthesized is assembled by site-specific genetic recombination. In addition to bringing together the separate gene segments and the C region exons of the antibody gene, these rearrangements also activate transcription from the gene promoter through changes in the relative positions of the enhancers and silencers acting on the promoter. Thus, a complete antibody chain can be synthesized only after the DNA has been rearranged. As we shall see, the process of joining gene segments contributes to the diversity of antigen binding sites in several ways. Each variable region is encoded by more than one gene segment. When genomic DNA sequences encoding C and V regions are first analyzed, it was found that a single region of DNA encodes the C region of an antibody chain, but two or more regions of DNA have to be assembled to encode each V region. Each light chain V region is encoded by a DNA sequence assembled from two gene segments, a long V chain segment and a short joining or J lines gene segment, not to be confused with the protein J chain, which is encoded elsewhere in the genome. The genetic mechanism involved in producing a human kappa light chain polypeptide from a C region exon and separate V region segments. Each heavy chain V region is encoded by a DNA sequence assembled from three gene segments, a V segment, a J segment, and a diversity segment or D gene segment. Then precise joining of gene segments greatly increase the diversity of V regions. During B cell development, the V and J gene segments for the light chain and the V, D, and J gene segments for the heavy chain are joined together to form a functional VL or VH region coding sequence by a process of site-specific recombination called VDJ joining. Conserved DNA sequences flank each gene segment and serve as recognition sites for the joining process, ensuring that only appropriate gene segments recombine. Thus, for example, V segment will always join to a J or D segment but not to another V segment. Joining is mediated by an enzyme complex called the VDJ recombinase. This complex contains two proteins that are specific to developing lymphocytes as well as enzymes that help repair damaged DNA in all our cells. So the lymphocyte specific proteins of VDJ recombinase are encoded by two closely linked genes called RAG1 and RAG2, RAG recombination activating genes. The RAG protein introduce double strand breaks at the flanking DNA sequence, and this is followed by a rejoining process that is mediated by both the RAG proteins and enzymes involved in general DNA double strand repair. Thus, if both RAG genes are artificially expressed in a fibroblast, the fibroblast is now able to rearrange experimentally introduced antibody gene segments just as a developing B cell normally does. So, moreover, individuals who are deficient in either RAG gene or in one of the general repair enzymes are highly susceptible to infection because they are unable to carry out VDJ joining and consequently do not have functional B or G T cells. T cells use the same recombinase to assemble the gene segments that encode their antigen-specific receptors. So, antigen-driven somatic hypermutation fine-tunes antibody responses. The control of VDJ joining ensures that B cells are monospecific. When activated by antigen, a B cell switches from making a membrane-bound antibody to making a secreted form of same antibody. B cells can switch the class of antibody they make. So, combinational joining of DNA segments. Junctional diversification during gene segment joining, combinational joining of L and H genes. The DNA rearrangement that occurs in class switch recombination. A B cell making an IgM antibody form an assembled VDJ DNA sequence is stimulated by antigen and the cytokines made by helper T cells to switch to making an IgG antibody. Then somatic hypermutation are four main mechanisms of antigen diversification. Those shaded in, bone marrow is there. So during B development is the bone marrow or fetal liver, while the mechanism shared antibody and helper T cells are also there. Then antigen antibody interaction. Next is about antigen antibody interaction. Antigen antibody interaction or antigen antibody reaction is a specific chemical interaction between antibodies produced by B cells of the white blood cells and antigens during immune reaction. The antigens and antibodies combine by a process called agglutination. It is the fundamental reaction in the body by which the body is protected from complex foreign molecules such as pathogens and their chemical toxins. In the blood, the antigens are specifically and with high affinity bound by antibodies to form an antigen-antibody complex. The immune complex is then transported to cellular systems where it can be destroyed or deactivated. And the specificity of the binding is due to specific chemical constitution of each antibody. The antigenic determinant or epitope is recognized by the paratype of the antibody situated at the variable region of the polypeptide gene. If one epitope is present, the antigen is monovalent. If both are one, it is called polyvalent or multivalent. A variable region in turn has higher variable regions which are unique amino acid sequences in each antibody. Antigens are bound to antibodies through weak and non-covalent interactions such as electrostatic interactions, hydrogen bonds, van der Waals forces, and hydrophobic interactions. Then affinity and avidity. Affinity measures the strength of interaction between an epitope and an antibody antigen binding site. Affinity measures the strength of interaction between an epitope and an antigen antibodies antigen binding site. Combined strength of total non-covalent interactions between single AG binding site of AB and single epitope is affinity of AB for that epitope. Then low affinity AB bind AG weakly and dissociates readily. High affinity AB bind AG tightly and remain bound closer longer. Then avidity, antibodies and antigens are multivalent, meaning they possess more than one binding site. The measure of the total binding strength of an antibody at every binding site is termed avidity. Avidity is also known as functional affinity. Avidity is determined by three factors: binding affinity, valency, structural rearrangement. Affinity versus avidity. Affinity refers to the strength of a single antibody antigen interaction. Each IgG antigen binding site typically has high affinity for its target. Then avidity refers to the strength of all interactions combined. IgM typically has low affinity antigen binding sites, but there are 10 of them, so avidity is high. Okay. Then major histocompatibility complex molecule. Every mammalian species studied to date possesses a tightly linked cluster of genes, the major histocompatibility complex or MHC, whose products play a role in intercellular recognition and in a discrimination between self and non-self. The MHC participates in the development of both humoral and cell-mediated immune responses. While antibodies may react with antigens and alone, most T cells recognize antigen only when it is combined with an MHC molecule. Furthermore, because MHC molecule act as antigen presenting cells, the particular set of MHC molecule expressed by an individual influence the repertoire of antigen to which that individual's Th and Tc cells can respond. The major histocompatibility complex is a collection of genes arrayed within a long continuous stretch of DNA on chromosome 6 in humans and on chromosome 17 on mice. The MHC is referred to as HLA complex in humans and as H2 complex in mice. Class 1 MHC genes encode glycoproteins expressed on the surface of nearly all nucleated cells. The major function of the class 1 gene products is presentation of peptide antigens to Tc cells. Class 2 MHC genes encode glycoproteins expressed primarily on antigen-presenting cells, macrophages, dendritic cells, and B cells, where they present processed antigenic peptides to Th cells. Class 3 MHC genes encode, in addition to the other products, various secreted proteins that have immune function, including components of the complement system and molecules involved in inflammation. Next, some other aspects related to class 1 MHC molecule. Class 1 MHC molecules contain a 45 kilodalton chain associated covalently with a 12 kDalton 2 microglobulin molecule. The chain is a transmembrane glycoprotein encoded by polymorphic genes within the A, B, and C regions of the human HLA complex and with the K and D by L regions of the mouse H2 complex class 1 MHC molecule. Beta-2 microglobulin is a protein encoded by a highly conserved gene located on a different chromosome. Association of the chain with the 2 microglobulin is required for expression of class 1 molecules on cell membrane. The chain is anchored in the plasma membrane by its hydrophobic transmembrane segment and hydrophilic cytoplasmic tail. Structural analysis have revealed that the chain of class 1 MHC molecule is organized into 3 external domains, each containing approximately 90 amino acids, a transmembrane domain of about 25 hydrophobic amino acids, followed by a short stretch of charged in the sense hydrophilic amino acids, and a cytoplasmic anchor segment of 30 amino acids. The beta-2 microglobulin is similar in size and organization to 3 domains. It does not contain a transmembrane region and is non-covalently bound to the class 1 glycoprotein. The next class 2 MHC molecule. Class 2 MHC molecule contains 2 different polypeptide genes, a 33 kilodalton chain and a 28 kilodalton gene, which associate with non-covalent interaction. Class 2 MHC molecules are membrane-bound glycoproteins that contain external domains, a transmembrane segment, and a cytoplasmic anchor segment. Each chain in a class 1 2 molecule contains 2 external domains 1 and 2 domains in one chain, 1 and 2 domains in the other. The membrane proximal 2 and 2 domains, like the membrane proximal 3 by 2 microglobulin domains of class 1 MHC molecule, pair sequences similarly to immunoglobulin fold structure. The membrane distal portion of a class 2 molecule is composed of the 1 and 2 domains and forms the antigen binding cleft for processed antigen. Class 1 molecule will be having membrane distal domains, membrane proximal domains. That means Ig folded structure, transmembrane segment will be there, beta 2 microglobulin, peptide binding cleft, class 2 molecule, etc. And the exon intron arrangement of class 1 and 2 genes reflects their domain structure. Separate exons encode each region of the class 1 and 2 proteins. Each of the mouse and human class 1 gene has a 5' leader exon encoding a short signal peptide followed by 5 or 6 exons encoding the chain of the class 1 molecule. The signal peptide serves to facilitate insertion of the chain into the endoplasmic reticulum and is removed by proteolytic enzymes in the endoplasmic reticulum after translation is completed. The next 3 exons encode the extracellular alpha 1, alpha 2 and alpha 3 domains, and the following downstream exon encode the transmembrane TM region. Finally, one of 2 3' terminal exons encode the cytoplasmic domain. Class 2 genes are organized into a series of exons and introns mirroring the domain structure of alpha and beta chains. Both alpha and beta chains encoding mouse and human class 2 MHC molecule have a leader exon, and alpha 1 or beta 1 exon, alpha 1 or beta 1 exon, a transmembrane exon, and one or more cytoplasmic exons. Then next is about antigen processing. Recognition of foreign protein antigens by a T cell requires that peptides derived from the antigen be displayed within the cleft of an MHC molecule on the membrane of a cell. So the formation of these peptide MHC complexes requires that a protein-protein antigen be degraded into peptides by a sequence of events called antigen processing. The degraded peptides then associate with MHC molecules within the cell interior, and the peptide MHC complexes are transported to the membrane where they are displayed, antigen presentation. So class 1 MHC molecules bind peptides derived from endogenous antigens that have been processed within the cytoplasm of the cell. Normal cellular proteins, tumor proteins, or viral and bacterial proteins produced within infected cells. So class 2 MHC molecules bind peptides derived from exogenous antigens that are internalized by phagocytosis or endocytosis and processed within the endocytic pathway. Then endogenous antigen is degraded by proteosomes. Peptide is transported to R via TAP. Then class 1 MHC alpha chain binds chylnexin. Then beta 2 microglobulin. Chylnexin dissociates. Chylreticulin, tapacin, and ERP57 bind. MHC captures peptides. Chaperones dissociate. Class 1 MHC peptide is transported from RER2 Golgi complex to plasma membrane. Then class 2 MHC alpha beta bind invariant chain blocking binding of endogenous antigen. Then MHC complex is routed through Golgi to endocytic pathway compartments. Invariant chain is degraded, leaving CLIP fragment. Exogenous antigen is taken up, degraded, routed to endocytic pathway compartments. HLA-DM mediates exchange of clip for antigenic peptide. Class 2 MHC peptide is transported to plasma membrane. Then next is about antigen presentation. T cells recognize foreign antigens in the form of short peptides that have been processed and displayed on the cell surface bound to MHC1 or MHC2 molecules. Antigens are often categorized according to whether they are derived from viruses, intracellular bacteria, or protozoan parasites. Endogenous pathogens, exogenous pathogens that replicate outside of the cell. Intracellular antigens are presented to T cells by any nucleated cell because MHC1 expression is ubiquitous. Then in contrast, exogenous antigens are taken up by professional APCs which process the antigens and present them in the context of MHC2, an important function of a professional APC. And the dendritic cell is to deliver a second signal co-stimulation to the T cell to alert it to the presence of infection. And endogenous antigens, including misfolded proteins and pathogen-derived peptides, are processed by the proteasome. This complex of protease typically generates peptides of 4 to 20 amino acids with a hydrophobic carboxy terminus. After trimming of the peptide by cytosolic protease, the antigenic peptides are translocated into the endoplasmic reticulum by the transporters associated with antigen processing. Meanwhile, a new MHC molecule is being synthesized in the endoplasmic reticulum. As it folds, it is found by calnexin, which is then replaced by calreticulin and B2. Then the new MHC1 molecule associates with MHC1 peptide loading complex. So tapsane physically links the MHC1 molecule and the TAP transporters. As the peptide enters from the cytosol, the cleft of the class 1 molecule receives it, and the peptide bound MHC1 molecule dissociates from the peptide loading complex and is recruited to the cell surface. Okay. So exogenous antigens are processed quite differently. Bacterial proteins are cleaved by proteases, cathepsins, and metalloproteases in the acid environment of the endocytic pathway. Meanwhile, MHC2 molecules assemble in the endoplasmic reticulum with another molecule called the invariant chain. The newly synthesized molecule transits the endoplasmic reticulum and the Golgi apparatus. After passing of the Li-loaded MHC2-DM complex through the Golgi into the late endosome, the invariant chain is cleaved by acid protease, leaving a residual peptide referred to as the class 2 associated invariant chain peptide in the MHC2 cleft. So CHIP occludes the MHC2 cleft and prevents peptides from loading until the molecule is in the lysosomal or late endosomal compartment containing the peptide. At that point, HLA-DM molecules remove CLIP from the cleft and stabilizes the molecule while the peptide is loaded into the cleft. So HLA-DM molecule can also facilitate this process in some settings. The fully assembled and loaded MHC molecule is recruited to the surface and serves to stimulate predominantly CD4 positive T cells. So thank you for listening. Next, we will be learning some other topic not related to this one.